Picture this. A scientist walks into a lab, stares at a flask of cloudy liquid, and says, "Inside this soup, there may be insulin, a malaria drug precursor, or the building blocks of tomorrow's materials." Hollywood hears that and says, "Excellent, add lightning, ominous violins, and a monster with 6 elbows." Reality is less dramatic, and somehow more impressive. Because synthetic biology is not mainly about building instant doom creatures that escape through air vents while a panicked intern drops a clipboard. It is, in many cases, about teaching microbes, tiny living factories, to make useful things on purpose. Drugs, chemicals, ingredients, specialized molecules, at industrial scale. That is not sci-fi. That is already mainstream biotechnology. Recombinant human insulin, for example, was approved in 1982 and became the first approved medical product derived from recombinant DNA technology. So tonight, we go deep into the rabbit hole. Not the monster in a vat rabbit hole, the real one. The one where biology becomes programmable. Yeast becomes a factory worker, bacteria become chemists, and humanity starts asking an ancient question in a new language. If life can be engineered, what exactly are we building? Part 1. The day humans stopped just finding nature and started editing it. For most of human history, we were collectors. We found useful things in nature, the way children find seashells, lucky, grateful, and mostly confused about how the ocean made them. A healing herb? Wonderful. A mold that kills bacteria? Amazing. A hormone hidden inside animal organs? Disturbing, but medically useful. We did not design biology. We harvested it. Then came the turning point. The realization that living cells are not mystical blobs. They are information systems. DNA is instruction. Cells read code. Proteins are built from those instructions, and if you can change the instructions, you can change the output. That is the quiet revolution underneath synthetic biology. Not playing God in the cartoon sense, more like learning that nature has been running the oldest software platform in the universe, and humans have finally opened the developer menu. And once that happened, an audacious thought emerged. What if instead of extracting rare molecules from plants, animals, or messy natural processes, we could engineer microbes to manufacture them for us? That idea became one of the defining engines of modern biotech, which, if you think about it, is both brilliant and humbling, because the future of advanced civilization may depend partly on persuading yeast to do paperwork. Part 2. Your first cyborg pharmacist was a microbe. Let's talk about insulin. Before recombinant human insulin, many patients relied on insulin purified from pig or cow pancreases. Then, researchers produced recombinant human insulin using microbes. And that approval in the early 1980s wasn't just a medical milestone, it was civilization changing its manufacturing philosophy. We went from find it in animal tissue to engineer microbes to produce the human version. A bacterium, an organism so small it could throw a party on the tip of a pin, became part of a global pharmaceutical supply chain. That's synthetic biology's secret superpower. It does not always arrive with fireworks. Sometimes it arrives as reliability, purity, scalability, consistency. And that may sound boring until you remember that boring in medicine often means millions of people can get what they need, with better control and less dependence on unstable supply methods. In other words, synthetic biology's first blockbuster was not a cinematic monster. It was a manufacturing miracle in a bottle. The villain of the story was not a mutant beast. It was inefficiency, which, to be fair, is a far more common threat to human life than tentacles. Part 3. The yeast that took on malaria. Now let's step into one of the most elegant stories in the field. Malaria treatment depends heavily on artemisinin-based therapies. Traditionally, artemisinin came from the sweet wormwood plant, but plant supply can fluctuate. Crops fail, harvests vary, timelines stretch, prices wobble like a shopping cart with one bad wheel. So, scientists pursued a radical alternative. Engineer yeast to make artemisinic acid, a precursor used to produce semi-synthetic artemisinin. And they succeeded. Think about the beauty of that. A drug associated with a plant, partially reborn through fermentation tanks, not because nature failed, but because biology could be extended. That's the crucial distinction many people miss. Synthetic biology is not often a war against nature. It is an effort to collaborate with nature, using design. To say, this pathway exists, this molecule matters, this process is too fragile. Can we rebuild it in a form that is more dependable? And when it works, the achievement is almost poetic. A disease that has haunted humanity for millennia meets a tool built from modern genetics, industrial chemistry, and microbial engineering. Ancient suffering, answered by programmable life, which is either inspiring or the plot of the classiest sci-fi film never made. Part 4. What synthetic biology actually is. Now, let's clear up the confusion, because synthetic biology sounds like someone is assembling a velociraptor from spare USB cables. But in practice, synthetic biology usually means combining genetic engineering, systems biology, and design principles to make organisms perform useful functions more predictably. In simple terms, you choose a biological job. You identify the genes or pathways involved. You insert, delete, tune, or rearrange them. Then you test whether the cell does the job better. That job could be making a therapeutic protein, producing a specialty chemical, sensing toxins, manufacturing ingredients, serving as a chassis for larger bioprocessing systems. So, no, most synthetic biologists are not trying to create instant monsters. They're more like coders, chemists, process engineers, and exhausted adults asking microbes to please stop misbehaving during scale-up. Because here's another truth the movies skip. Real biology is messy. Cells mutate, pathways interfere, metabolism reroutes, yields collapse. A strain that behaves beautifully in a lab can become dramatically less impressive when asked to perform in a giant industrial tank. Synthetic biology is powerful precisely because living systems are hard to control, not because they're easy. This is not magic. This is engineering in the most irritatingly biological environment imaginable. Imagine programming a factory where the machines reproduce, improvise, get stressed, and occasionally decide your plan was merely a suggestion. That is synthetic biology. Part 5. Why Instant Monster is Bad Science and Bad Storytelling. Let's address the myth directly. The public imagination often leaps from engineered microbe to overnight abomination, but that fantasy misunderstands both biology and time. A microbe engineered to produce a drug or chemical is usually optimized for a narrow, highly specific function under controlled conditions. Commercial synthetic biology is built around constrained systems, production targets, containment practices, and quality control, not spontaneous cinematic chaos. The organism is not trying to become a supervillain.
It is trying, usually with some reluctance, to metabolize feedstock and keep its internal chemistry from collapsing. That is not the same as instant monster. And honestly, the more plausible horror story is not the bacteria became a dragon. It's the batch failed on Friday, the contamination report came on Saturday, and the investor call is on Monday. That is the biotech thriller nobody asked for, but every operations team understands. Still, the myth exists for a reason, because deep down, people sense something true. Once humans can redesign living systems, the stakes change. And that instinct is not foolish, it is morally alert. The challenge is to separate realistic risk from cartoon panic, because if we fear the wrong
and if we romanticize the science, we become reckless. Part 6. The real risks aren't tentacles, they're power, scale, and consequences. The real risks in synthetic biology are not usually instant monster scenarios. They are more serious and less theatrical. Unintended ecological effects, misuse, inadequate containment, poor governance, supply chain dependence, and the gap between technical capability and ethical maturity. That concern is not hypothetical. In late 2024, a group of scientists publicly warned that creating mirror-life microbes could pose severe, unprecedented biosafety risks if such organisms were ever developed and released. That warning proves the central point of this video. Synthetic biology is already real enough and powerful enough that serious scientists debate boundaries before certain lines are crossed. That is what grown-up science looks like. Not denial, not hysteria, deliberation. And this matters because humanity has a recurring habit. We invent a tool, we celebrate the breakthrough, we scale the tool. Then, somewhere between profit and prestige, we discover we were much better at building than governing. We did it with fossil fuels, we did it with social media, we did it with financial leverage. And yes, if we are careless, we can do it with biology. So the right posture is neither fear everything nor trust everything. It is disciplined awe. Synthetic biology deserves wonder. It also deserves oversight. Because once biology becomes a platform technology, the question is no longer whether we can engineer life for useful ends. The question becomes who decides what counts as useful. Part 8. The factory of the future may look alive. Now zoom out. What happens when engineered microbes become normal industrial tools? A profound shift. Factories begin to look less like smoke-belching monuments and more like controlled ecosystems. Instead of forcing matter through extreme heat and harsh chemistry alone, we increasingly ask cells to assemble molecules through metabolism. Feed them sugar, carbon sources, nutrients, and instructions, then let biochemistry do what metallurgy cannot do as elegantly: build complexity from the inside out. This is why experts describe synthetic biology as relevant not just to pharma, but to chemicals, materials, food, and energy-adjacent bioproducts. Commercially available synthetic biology products already span multiple sectors. In that future, the line between manufacturer and cultivator starts to blur. You may not simply run a plant, you may grow one. Not tomatoes, outputs. That is a conceptual earthquake, because the industrial age taught humanity to dominate matter. Synthetic biology teaches humanity to negotiate with living systems, and negotiation is trickier. Steel does not mutate. A furnace does not evolve. A bacterium absolutely will surprise you, if given enough time, enough pressure, and a bad enough incentive structure. Which is why synthetic biology's rise may produce a new kind of industrial wisdom, not brute force, stewardship. Part 9. The philosophical plot twist. Here is where the rabbit hole gets truly deep. Synthetic biology makes us confront an uncomfortable possibility. Life is not only sacred,
it is also functional. Not only sacred, don't miss that distinction, but functional enough to be engineered, optimized, modularized, and repurposed in limited ways. That realization can provoke two opposite reactions. One person hears it and says, then life is nothing but machinery. Another hears it and says, then the machinery is far more astonishing than we imagined. The second view is wiser, because discovering that biology has mechanisms does not reduce wonder, it relocates wonder. The miracle is not lost because we understand more. The miracle gets sharper. A cell is not less marvelous because we can map pathways inside it. If anything, it becomes more marvelous. You are looking at something that stores information, reads instructions, repairs itself, adapts to stress, and can be coaxed sometimes into becoming a pharmaceutical workforce. That is outrageous. That is sublime. That is also the point where science and humility should shake hands. Because the more we can edit life, the more we must resist the delusion that we fully comprehend it. Power grows faster than wisdom, unless wisdom is trained on purpose. And every technological age eventually reveals the same moral equation. Capability without character scales disaster. Part 10. What this means for the next 20 years. Over the next 20 decades, synthetic biology is likely to become even less visible and more important. That is how transformative infrastructure usually works. You don't wake up each morning whispering, "Thank you, semiconductor fabrication," yet semiconductors shape nearly every modern action. Synthetic biology may follow a similar pattern. You may not see the microbe, you may not know the pathway, you may never hear the strain name, but the medicine, ingredient, material, or process may already have passed through a living factory somewhere along the chain. That trajectory is consistent with how the field is already described by scientific reviews and government research programs. A platform for scalable production across biomedicine and bioproducts, not a fringe curiosity. And that means the public conversation needs to mature. Less, did scientists create a blob? More, what standards govern engineered organisms? What containment is required? Who benefits economically? Which applications solve real human problems? Where do we draw hard lines? Because the future will not be shaped only by what labs discover. It will be shaped by what societies permit, incentivize, regulate, and reward. Technology doesn't enter history alone. It arrives handcuffed to politics, money, ethics, and human appetite. Always. So the right posture is neither fear everything nor trust everything. It is disciplined awe. Synthetic biology deserves wonder. It also deserves oversight. Because once biology becomes a platform technology, the question is no longer whether we can engineer life for useful ends. The question becomes who decides what counts as useful. The ending, no monster, just a mirror. So let's end where we began. Synthetic biology is real. Engineered microbes making drugs and chemicals is not speculative fantasy. It is already part of mainstream biotech. Recombinant insulin and semi-synthetic artemisinin are the two clearest historical examples. And no, that does not mean tomorrow morning a giant lab beast will kick down your door and demand venture funding. The real story is stranger than that and deeper. We have entered an era where the smallest forms of life can be redesigned to extend human capability. A bacterium can become a manufacturer. A yeast cell can stabilize supply for vital medicine. A gene pathway can be treated like a production line. A flask can become a factory prototype. That is not the birth of an instant monster. It is the birth of a new relationship between intelligence and life. And like every powerful relationship, it reveals us, our discipline, our greed, our mercy, our impatience, our imagination, our restraint.
Synthetic biology is not just a tool for remaking microbes, it is a mirror for the species using it. And the real question is not whether we can engineer cells. The real question is whether we can engineer a civilization wise enough to deserve what those cells can do. Because the future may not roar, it may ferment. And if that doesn't sound dramatic enough, you've clearly never met biology. If you want more videos where we cut through the hype, chase the real science, and go all the way down the rabbit hole without losing the trail, subscribe now, because the next revolution may already be growing in a tank somewhere. And trust me, the story is just getting started.